Welcome to all. This is Ganapathy, Assistant Professor, PG and Research Department of Microbiology, JJ College of Arts and Science. Today we discuss about the topic TCR gene rearrangement, which means T cell receptor. The germline organization of the human T cell receptor alpha and beta loci. The arrangement of the gene segments resembles that at the immunoglobulin loci with separate variable diversity joining gene segments and constant genes. The TCR alpha locus consists of 70 to 80 variable alpha gene segments, each produced by an exon encoding the leader sequence. How many of these variable alpha gene segments are functional is not known exactly. A cluster of 61 G alpha gene segments is located a considerable distance from the variable gene segments. The J gene segments are followed by a single C gene which contains separate exons for the constant and hinge domains and a single exon encoding the transmembrane. Each TCR beta C gene has separate exons encoding the constant domain, the hinge, the transmembrane region and the cytoplasmic region. The TCR alpha locus is interrupted between the J and V gene segments by another T cell receptor. Locus, the TCR delta locus, not shown here. The first one is TCR alpha chain and the second one is TCR beta chain. Generation of diversity in the TCR. Combinational diversity, multiple germline segments. In the human TCR, variable segments 70 to 52. Diversity segments 0 to. Joining J segments 61 13. The need of two pair and chains to form a binding site doubles the potential of diversity. TCR rearrangement is very similar to IG rearrangement. Somatic recombination, combinatorial diversity. Which is TCR alpha gene rearrangement rescue pathway. Lymphocyte activation, proliferation and differentiation. The adaptive immune system relies on both specific antigen recognition and on the ability of lymphocytes to maintain a memory of previous encounters with antigens. These two features represent the essential basis of required immunity. The capacity of the immune system to recognize billions of epitopes is established via the complex process of antigen receptor gene recombination that produces countless combinations of specificities for foreign antigens. T cell activation and differentiation. Mature T cells exit the thymus with all the genetic components needed to recognize antigens. However, what these naive T cells lack is the ability to respond rapidly to their first encounter with the antigens recognized by their specific TCRs. During a productive immune response, when naive T cells are first activated, they require correct antigen representation over an extended period of time as they undergo the complex process of blast cell transformation. During this process, they convert from small quiescent cells to large, highly proliferative cells. Recently, activated T blast cells and differentiated T cells remain tightly regulated and rely on ongoing activation of T cell receptor signaling to express inducible immune response genes. The development of T cells. Humoral and cell mediated immunity. The humoral and cell-mediated branches of the immune response. The humoral branch comprises lymphocytes of the B cell lineage. Antibodies are the effector molecules produced by this response. The process begins with the interaction of B cell receptors BCRs with antigens. Binding of the antigen promotes differentiation into antibody secreting cells. B cell maturation. During maturation, B cells again gain antigen receptor molecules termed B cell receptors, which are displayed in large numbers extracellularly on their membrane. These membrane-bound protein complexes contain antibodies which enable specific antigen recognition. Each B cell initially produced as only one kind of antibody, which makes every B cell unique. It is the immense number of B cells in the body, each of which produces a unique antibody that allows the immune system to detect such a wide variety of pathogenic antigens. B cells containing antibodies that recognize self antigens are destroyed before they can mature, preventing the immune system from attacking the host. Once B cells mature in the bone marrow, they migrate to lymph nodes or other lymphatic organs where they may begin to encounter pathogens. End of the session. Thank you.